This video is part 2 of the colon cancer laboratory, in which I will describe the individual slides and indicate the structures of interest. As you look at these slides, I would like you to keep in mind the staging of colon cancers by observing the landmarks which they have passed. The first thing I want you to do is become familiar with the normal structure of the colon wall. For this, I ask that you look at the slide labeled colon 4. As indicated here, there is a small region of normal colon wall. This is the region I want you to look at first. The rest of the section contains adenocarcinoma, but you will examine this later. Examine the normal region of colon 4 under the 4x objective lens. You should be able to identify the 4 major layers of the colon wall. Starting from the bottom and going to the top, the first layer is the serosa that contains blood vessels and adipose or fat tissue. Second comes the muscularis externa or muscularis propria. This consists of 2 layers of smooth muscle oriented at right angles to each other, the circular and the longitudinal. Although difficult to observe at this magnification, between these 2 layers are ganglions, which are collections of neuronal cell bodies. The third layer is a submucosa, which contains a loose connective tissue and blood vessels. This layer generally stains slightly lighter than the smooth muscles which are located on either side. And finally, the innermost layer is the mucosa. Now, within this mucosa, locate the following structures. The muscularis mucosae, which is a line of smooth muscle that marks the boundary of the mucosa. Do not confuse this with the outer and larger muscularis externa, which is also composed of smooth muscle. Next, identify the lamina propria, or loose connective tissue under the epithelium. And finally, the lining epithelium, that is a simple columnar epithelium and contains numerous mucous goblet cells. Also, locate the crypts of Lieberkühn. Now place the slide labeled colon 1 on a white piece of paper and examine it without magnification. Note that it contains 2 thin sections. The bottom section is somewhat better preserved than the top, so I will concentrate on it. The eosinophilic, that is orange, region is the smooth muscle of the muscularis externa. The thin basophilic or blue band is the normal mucosa, while the mushroom-shaped structure extending from its surface is the polyp. Examine the polyp under the 4x objective lens. Locate the muscularis mucosae and note that the polyp does not extend beyond it, although the muscularis is broken in regions by lymph nodules. This means that the polyp is non-malignant or benign. Center the slide over the junction between the normal mucosa and the edge of the polyp, indicated here by the yellow asterisk. Switch to the 10x objective lens so that you can simultaneously view both the normal mucosa and the polyp. In some regions of the polyp, the epithelial cells are more basophilic than the normal epithelium and contain fewer goblet cells. These characteristics indicate that the epithelium shows some degree of dysplasia, and for this reason, the polyp is considered to be adenomatous. Now switch to the 40x objective lens. This view shows a side-by-side comparison of the normal epithelium on the right and the adenomatous epithelium on the left. At this magnification, you can better see the differences between the normal mucosa and the adenomatous polyp. Clearly, there are fewer goblet cells. The cytoplasm is darker staining. The nuclei are larger and more elongated, and the nuclei are arranged more randomly than those in the normal epithelium, which are lined up in straight rows. Also, examine the lamina propria of the polyp. It contains dilated blood vessels and numerous eosinophils, a type of white blood cell. View the slide labeled colon 2 by eye. Even without magnification, you should be able to see a number of polyps, which often occur in groups. Also locate the presence of large cyst-like structures in the muscularis externa that are filled with clear mucin. And finally, locate the region of normal colon tissue located on the edge of this section. Place a slide on the microscope and examine the region indicated by the red asterisk using the 4x objective lens. This is a low magnification view of the slide showing a number of different structures of interest. There is the normal mucosa located on the top. On the top left, this merges with a villus polyp. And on the bottom, you can see the muscularis externa that contains adenocarcinoma located in cyst-like structures. Examine the polyp under the microscope and observe that the outer regions consist of long finger-like projections which define this structure as a villus polyp. This is a high-power view of the cells lining one of the villus polyps. The lining epithelial cells have an adenomatous character in that they are more basophilic and contain fewer goblet cells than the normal epithelium. Switch back to the 4x objective lens and examine the cysts located in the muscularis externa. Note that some of these cysts contain polyp-like structures that are composed of adenocarcinoma. This is another low magnification view of the adenocarcinoma located in the muscularis externa. These cells are responsible for making the clear mucin that fills the cysts. With regard to staging of this colon cancer, it has clearly penetrated the muscularis externa, but I could find no evidence that it had passed into the serosa, and on this basis I would give it a stage of T2. Switch to the higher power and examine the adenocarcinoma lining these cysts. Note that these tumor cells contain relatively few goblet cells and are darker staining. Also, the nuclei of the tumor cells are larger, more heterochromatic, have more prominent nucleoli than the normal epithelium. This is indicative of a higher rate of transcription. Visually examine the slide labeled colon 3 and note that the extent of the tumor indicated by the blue stain is much greater than in the previous slide. Also observe that there is a light staining adipose tissue of the serosa on one side. On the other side of this section is a small region of normal mucosa. Please make a note of its location. Place the slide on the microscope and examine the normal mucosa under the 4x objective lens. Observe that the mucosa and submucosa are relatively normal. However, below this, the muscularis externa contains adenocarcinoma. Follow the normal mucosa around the edge and note that it transitions into a villus adenoma in which the epithelium is clearly not normal. It lacks goblet cells and is in the form of long finger-like projections, giving it a villus character. Below this region, the adenocarcinoma has invaded past the submucosa into the muscularis externa. This is another view of the muscularis externa containing a nodule of adenocarcinoma. The cells here are still forming gland-like structures. Next to this region is a serosa composed of adipose connective tissue. Now in this view, the adenocarcinoma appears to be right on the borderline between the muscularis externa and the serosa. In other views, it has clearly penetrated the serosa, and on this basis would be given a stage of T3. This is a high-power view of the adenocarcinoma cells. Note that they are still forming gland-like structures. It is one cell layer thick based upon the location of the nuclei. It lacks goblet cells, and there are several mitotic figures indicating rapid growth. Examine the slide labeled colon 4 by eye and identify the region of normal tissue on one end. Observe that the bulk of the tissue consists of adenocarcinoma that has resulted in surface ulceration. This section does not contain any residual adenoma. Scan the slide under low magnification. This is the normal region that you had examined at the beginning of the lab. Move the slide to a region of the serosa and observe that the adenocarcinoma has invaded through the muscularis externa and penetrated into the serosal fat layer. This colon cancer would be staged as T3. Switch to higher power and examine the adenocarcinoma. Shown here are the different regions of the adenocarcinoma. On the left is a region in which the tumor cells are still forming gland-like structures with lumens, and it's considered to be well differentiated. On the right, the tumor cells have lost some of their differentiated character in that much of the glandular-like structures have been lost. This region would be considered to be moderately differentiated. Examine the slide labeled colon 3 by eye and note that the tissue is broken in one region. There is a small segment of normal mucosa on one end. Also observe the location of the large cyst-like structures in the muscularis externa. And finally, locate the ink that defines one of the surgical margins next to the serosa. Place the slide on the microscope and center it over the normal mucosa. Under low power, observe that immediately beneath the normal mucosa there are nests of adenocarcinoma in both the submucosa and the muscularis externa. Now move the slide to the region near the ink. In this location you will observe cysts that are lined by adenocarcinoma. The tumor cells have clearly invaded the serosa, and consequently this would be given a stage of T3. Examine one of the cysts under high power. You can make out the light-staining mucin within the cyst that is produced by the adenocarcinoma. This indicates that this is a mucinous type of adenocarcinoma, which has a slightly worse prognosis than its non-mucinous counterpart. Observe the slide labeled colon 6, which contains lymph nodes taken from a colon cancer patient. By eye, note that there are 5 and possibly 6 lymph nodes on the section. The nodes tend to be basophilic. Sometimes the nodes are difficult for the surgeon to identify. Although you cannot tell this without magnification, 3 of the nodes are normal, while 2 on the right contain metastasis of colon cancer. Examine one of the nodes that contains metastasis under low power. Some of the node is normal and composed primarily of small lymphocytes that form nodules and lighter staining germinal centers. On the left side is a single mass of metastatic cancer from the colon. This is much larger and clearly distinct from the rest of the node. Switch to higher power and observe the metastasis. It is a continuous mass and still retains some of its epithelial character. It is also rather homogeneous. The cells are large, with large nuclei, easily distinguished from the surrounding normal lymphocytes, which resemble small dots.